Hello everyone. My name is Patrick Werner. I'm happy to be here. Thanks for the invitation and the possibility to speak about genomics today. I'm one of the co-chairs of the Clinical Genomics workgroup of HL7 International, and we designed an implementation guide specifically for genomic reporting, and I will talk about this today. Myself, I'm doing FHIR for quite some time, started pre-DS2.2. I'm doing a little bit of contribution to the Happy server from time to time, a little bit FHIR core code, if I have some spare time. So our learning objectives for today is the FHIR Genomics Motivation of the IG and the significance of it, the tools of the FHIR Genomics group, the IG structure, the genomics reporting IG profiles. We will have a look at those. Some examples so you can see how an actual instance of these profiles would look if you would implement it. I will have a brief look at tools which are adopting or using our IG as a data model or which are compliant to this data model. And at the end, we have a short review and close session where you have the opportunity to ask questions. Yeah, so first, the FHIR Genomics Motivation. The problem was the goal is to modernize genomic lab reports. We want to enable modern big data approaches, which is hard if you have data stored as a PDF or even faxed. We want to enable clinical decision support systems because genomic data, or especially how's it called? Sorry, precision medicine approaches. You have to take into account all the observations, all the variants of the patient in order to be able to determine the best medication or treatment for the patient. So the physicians in the hospitals, in the day-by-day care, they are overwhelmed with a lot of data, which is too much to handle by hand and look up relevant pathways and medications. And we also want to facilitate downstream research and reanalysis. So if you want to do analysis of all your cases you have in your hospital, if they are stored in the PDF format, it's getting really hard, I guess. So the Oncology Report is a transaction bundle, contains patient, practitioner. No, sorry, this slide doesn't belong here. I will come back to this, sorry. So yeah, coming back to the nowadays genomic reports. It's currently PDFs, sometimes even via fax, so it's super hard to handle it by a computer or do an automated interpretation of it. Of course, this is not machine readable. You could argue, okay, if I have a PDF, I could extract something from the PDF, that's right. But unfortunately, most of the PDFs I saw, they weren't even OCR'd because in the lab, they are creating a PDF for the report, then they're printed out, then it's signed by the responsible lab personnel, then it's scanned into the computer again, sent via email to the hospital. In the hospital, the secretary of the physician prints it out, gives it to the physician, he signs it, scans in to the system again, and we are losing all the structure, and it's getting harder and harder to OCR. And the structure changes from lab to lab, of course. And it requires expert knowledge or additional knowledge to read or to do the interpretation on them. So our solution to this is structured data, of course, FHIR. So precision medicine, I talked about it in the introduction. It's an emerging approach for disease treatment. So oncology back in the days was totally focused on the position of the cancer, of the tumor. So this is a liver tumor, this is a brain tumor, and this body position determined the treatment. But nowadays we shifted and we are now looking at the individual gene variants of this patient. So it's getting less important to know the location of the tumor because sometimes tumors for different body location, for example, black skin tumor and some liver tumors, they share the same cause, the same genetic problems. So for that, we need the deeper genetic analysis. And And also an emerging approach in the clinical care are tumor boards or molecular tumor boards, having a look at these reports. And I've been in contact with some of these tumor boards in some hospitals, and it takes a huge workload. In some hospitals, per patient, they invested almost 20 to 30 minutes to prepare the tumor board, to gather all the information, put it together into a PowerPoint presentation, and then have a look at it. And if you have 10 patients per board and 20 to 30 minutes per patient, we don't have enough physicians to cover that amount of work. So this needs automation as well. We want to enable point-of-care apps for precision medicine. So if you are point-of-care, I'm not really happy about the term maybe, because it's point-of-care, I'm thinking about bedside. I think the primary goal is not to discuss the variant with the patient on the bedside and the possible treatments, which could be cool to have as a patient. I would like that, but not every patient will be that deeply involved in his treatment. So I would say it's more like for precision medicine or for virtual tumor boards, it's almost the same. If we have structured genomic data, we can access to this data, and we can do an integration into our EMR via clinical decision hooks or Smart on FHIR, access this data and join it together with publicly available data sources like OncoKB or CBioBoard portal. The problem with these knowledge sources, they are not FHIR compliant for sure. So this will be one of the next problems, but not part of this presentation. How do we integrate external knowledge sources if they are so diverse, so heterogeneous, if they have such different interfaces? Yeah, for the virtual tumor board approach, so this is one of our solutions we do at the Molit Institute where I'm working. We have an open source virtual tumor board software called V2. And there's a manual request for now to the lab. We will shift that to some business process supported workflow engine to do electronic requests. But for now, it's a manual request, but we are getting kind of formatted data back. So it's TSV or CSV with tab separation data in an enclosed format. It's nothing common. It's a very specific format to this lab. And we do the transformation to the FHIR IG resources at the first place. Then we have it in our FHIR server inside of the hospital, and then we can access this data and render it during the tumor board. So I hope it's a little bit on the small side, but here you can see this is a list of variants of a patient read from the FHIR server and then displayed to all the participants of the tumor board so they can have a discussion about the correct treatment of the patient. And here to the far right, the CBio portal link, if they click there, they end up in the CBio portal at the right position, like in the CBio portal at the page which is talking about this variant, which could be easily extended to other knowledge sources, which we are currently doing. Yeah, so back to the point of care, all for the tumor board is basically the same. We are linking the patient-specific data to reference knowledge in the best case to give additional guidance to the physicians and create even more knowledge or information at least. And we can, for a single patient, show the information of the variants. We can show the most common mutations in similar cancer populations to have a look. So this is what CBio portal is actually doing. You can have a look at another population from a study which has similar variants and then see the outcomes and discuss if a similar treatment for your patient would be the right way to go. We also, or you could also facilitate downstream research and analysis by creating anonymized case databases with a FHIR interface, so basically storing all the reports inside of the FHIR database. It doesn't have to be pure FHIR, so we had use cases where they built practically a facade. So the actual data was G4GH standard formatted. Then they built a facade to serve this in a FHIR format to be able to consume it in a smart and FHIR or CDS hooks app. For this, we need all the variants, so the genomic report, but as well some additional case data about the patient, his diagnosis, his TNM status, etc., and outcomes of the patient about the treatment, if the treatment was successful or not. And if we're doing that, then we can start doing some machine learning, big data approaches to get a deeper understanding on how to treat different cancer variant profiles. So this facade for the G4GH variant was highlighted in the May 2019 HL7 newsletter, if you're interested. They called it GACS, General Archiving and Communications, Genetic or Genomic Archiving Communication System, like PACS. I'm not sure if I like the term, but yeah, there's a GACS. If somebody says GACS, it's something with genomics. That's a takeaway. So this is how they built this thing. Yeah, you can see that. They had a Cerner-based EHR connected to a CDS hook server here, serving the patient data. But the actual variant data is retrieved through a data retrieval service, which then, so this would be the GACS, which was done by Intermountain in this case, serves the variant data as a FHIR resource. This is consumed by the CDS hook server. Some pharmacogenomic rule engine was involved as well, and then they can give back an information card to the physician inside of its normal Cerner EMR, but with additional information and with data which maybe couldn't be captured inside of the EHR or EMR. So in Germany, for example, if I talk to physicians and ask them, could you capture structured genomic data inside of your EHR? They always say no. So there's a need for an additional server storing all this genomic information for now, and as long we don't have the opportunity to have like EHRs speaking FHIR really nice and to be able to support this genomic profiles as well. Yeah, coming to the FHIR genomic tools. So we are the clinical genomic workgroups. I noted down some important links. So this is the Confluence links to our workgroup. If you're interested, you can have a look there. There are all the agenda items and stuff like this. Chatfire.org. If you don't have a Zulip account, you should get one. It's the most important resource if you're working with FHIR. We have our own stream called Genomics. Feel free to ask your questions there or present your use cases. We are always interested in feedback about our IG because we are doing this for so long, we are getting kind of blind, and it's good to have external input about the good things and about the stuff which doesn't work to further improve our IG. And we have the mailing list, the clinical genomics mailing list, ClinGenomics at lists at lists.hl7.org. Yeah, but if you have a Zulip account, go for the Zulip one, it's much faster. We also have a domain analysis model created by Clinical Genomics Workgroup in 2018 as a base for the IG. And we have our implementation guide, Genomics Reporting, which is still a draft. We are really currently publishing the first stable version, the STU1 version, but there are some publishing problems in the publisher somewhere. So we have to go for the publication, but now we're waiting for the publication to happen. Yeah, where could you find this implementation guide? The easiest is Google FHIR Genomics Reporting. It should be quite high. Or you could go to fhir.org/guide/registry and search for genomics. Or you go to hl7org/FHIR/genomics, and it's linked from there as well. The actual link to the Genomics IG is on the last slide of this presentation. So our implementation guide covers all aspects of genetic reporting for humans. So that's the goal. We are not sure if we already reach it. I doubt it, but yeah, that's the goal. We want to have a representation of known variants, mainly using terminologies like a cosmic ID. So we know this variant, it's a described variant in the genomic space, so we could just point to the defining ID of this variant. We want to capture the relevance of these identified variations in the terms of disease pathology, pharmacogenomics, transplants suitability, which is a really special case in genomics. These are the guys from the bone marrow donor program. They're working inside of the work group. One of our co-chairs is actually from there, Bob Milius. And they are always interested only in one gene, HLA, because they're doing HLA typing. And we want to support full and partial DNA sequencing, including whole genome or exome studies. The focus for now is on the data structures for encoding the genomic test results. So we are seeing us as building block builders. We have a lot of profiles which are there to be consumed or used by other implementers which have more specific use cases. So our profiles don't have must supports. We have very weak bindings to terminology, if possible, to be able that downstream usage is easy. So if we are defining, at the beginning we had a lot of must support items there. And MCode was one of the first IG which were using or trying to use our profiles, and they couldn't because we had too many must supports. So we got rid of this. For now, this is just building blocks. If you do something with genomics, have a look and use the profiles or derive from the profiles you like. Yeah, we are internationally or international in scope. That means that our code systems are not protected by any licensing fees, so unfortunately no SNOMED. We make heavy usage of observation diagnostic report. Almost everything is an observation, and the diagnostic report encapsulates all these informations. We avoid pre-coordinating the type of the variant, so we are capturing all the details in distinct component fields. So observation component is also very heavily used in the IG. We allowed for variability in the amount of discrete information captured. So we are not enforcing too much by 1 to something cardinalities. You use the parts you like to use. Yeah, and we are trying to be aligned with the version 2 implementation guide of the genomics as far as it's possible. It's quite aligned. So all the line codes in version 2 genomic reporting, which are qualifying the kind of observation you are doing, are the same in FHIR or in version 2. Our IG looks something like that. We have the table of contents like every IG. There you can see the different topics we are talking about in the IG, the background, how to do reporting in general as a report, how you do variant reporting, some glossary, because we identified some people have differing understanding of different concepts or values of concepts or names of concepts. Pharmacogenomic reporting, somatic reporting for the cancer oncology space. Histocompatibility, conversion from SU2, I think that's gone for now. A relation to version 2, etc. Just have a look. Coming to the actual report. So the genomics report, its category is genetics. The code for genetics is 81247-9 in long. There's one extension, a related artifact, that you can point to related artifacts in the sense of my decision is based on this knowledge source. So you can put in a DOI or something to a paper or to some other resources which have contain knowledge, basically. We support media attachments so that you can also include some histologic pictures, for example, or the PDF report if you like to. you can reference sub-reports, and we have the different results, so our main building blocks of the observations. So we have the region studied, which signifies what was looked at, because if you just get the results, it's also very important to know what was ordered or what was looked at, which could sometimes differ. The actual findings, so variants, named genotypes, etc. Implications, these variants from the patient, what's the implication on this in terms of medication efficacy, pathogenicity, and so on. There's an overall interpretation, and yeah, this is the link where you can go to this overview. So all the IG in a simple form is this. All the green stuff are observations. The yellow ones are other resources. The diagnostic report is another green. Yeah, so I talked about the variant. Derived from the variant, we will have the genotype, the haplotype. We have a structural genetic finding, which will be put into the variant soon, I guess. The sequence phase relationship points to two variants to define the sequence phase relationship. The report, overall interpretation, I just talked about it, region studied, and you can group different observations by a thing we call grouper, because physicians like to have lists. I want to have all single nucleotide variants in a list. So we created a grouper to be able to group it. Implications, so this is currently ongoing work. This is how it will be published, but already we know this will be changed, because as you can see, that's quite a lot of profiles for impact. And we are trying to squeeze these together. For example, diagnostic impact, prognostic and predictive impact, they could be just one cancer, somatic, what you like call it, impact with a qualifier if it's diagnostic, prognostic, or predictive. So this will come into the next version. If you have a look at the continuous build of our IG, you will notice that there are still changes, but now we are publishing our first stable version. Yeah, basically to have a stable version, because people don't like to, of course, implement changing IGs. So we need some frozen states. Yep. So this is the report result references. We have the genomic finding on top, which is a new thing, I guess. Everything with a red line outline is an abstract profile, so it's not implemented, it's just derived from four other profiles. Genomic implication has a recommended action as a recent reference pointing to it. So there's the recommended action. This is really the thing you are capturing the recommended treatment for the patient. And also some recommended follow-up if you think this patient needs a follow-up visit or some diagnostics, you can express it here. Yeah, so the linkage is done through observation has member or observation derived from, depending on the direction or the meaning of the connection between the different resources. Yeah, we have some other resources. Of course, the request for genetic tests, which is a service request, profile service request. We have the specimen, which is sent to the lab. We have the patient, of course, and organization or practitioner or group which are performing the genomics report or which are the requester for the recommended action at the end. We also have this supporting information. So as I said before, it's not just about the variants to be able to create some recommendations for the patient. It's not just the variants, it's also some background about the patient, which sometimes also needs to be sent to the lab. Because in the lab, at least in our use case, in our institute, in the lab, there are the geneticists, and therefore they need this supporting information with the specimen. So there's something called family member history, which is the history of the family members, basically, my uncle had some cancer when he was 40 and died from it or something like that. Risk assessment, was there a higher cancer risk assessed? Some document references to other findings, other reports which are not in a structured way or are in a structured way. And we have, of course, other clinical observations like FISH tests and stuff, which is not NGS, but immunohistology, I think in English, sorry. Yeah. So now we're coming to the most interesting thing, I guess, in my opinion, which are the findings, the actual variants. We have different profiles of the findings. We have the variant, the most used one. We have haplotype and genotypes, and a sequence relation, sequence phase relation, which is derived from either a molecular sequence, an haplotype, or a variant. This is how it looks, the abstract profile. So again, this was the one with the red surrounding. It's an observation derived from genomics base, but it's adding something. It adds the gene studied ID as a component. So yeah, I told you we are using components a lot, and we're using them because it's a handy, like a key value store. So you have the qualifier of this component. This component has the code 48018-6, which signifies stands for the gene studied ID, and then the actual value is bound to HGNC. So you have some HGNC ID about the idea about the gene you studied. You want to know the location on which chromosome. It's another component, and you want to know the reference sequence assembly. What is maybe missing here is the not the chromosome location, the exome location. I got some use cases, they really insisted that they wanted to know the exome location. So this is something to add in the future, probably. Yeah, codable concepts, codings and text. We are using them a lot because all this component, key value store thing, we want to qualify everything in a semantical way. There are different possibilities. Of course, you could have a text-only codable concept, which is a bad thing to have, I guess. So this would be the simplest one, AC, add, snip. You could have it coded in different code systems, and you, of course, it's a value codable concept. You could combine the different codings so that you use GL string and FHIR together, which doesn't make sense, but you get the idea. So the variant profile is huge. A lot of components, because it's a lot of information. So if you have a look at the diagnostic report, we got some report. So the hospital we are working with, they tried a new genomic lab, and the report was 30 pages long. So there's a reason why we are having so many components. Important thing is the method. How was this variant determined? Next level, next generation sequencing or FISH test or whatever, some probe testing. We have the value codable concept genetic variant assessment, which is bound to positive, negative, no call, and indeterminate. So that's the actual result. At this gene, is there something? Yes, no, the most simple way. And then we have additional information, the variation code, the DB SNP ID, the change in HGVS for coded or genomic HGVS, the change type, was it a substitution, frame shift, same for amino acid changes. Then the coordinate system, which was used, like 0, interval-based, 1-based. The start end for the change. The alt allele, the ref allele, so A was substituted by C, for example. The ref sequence assembly. The copy number, the allelic state, read depth, allelic frequency, the source class, which is like, is this a structural variant or is it a single nucleotide variant, inner start end, outer start end, so I can say I have a change in the DNA and I'm not sure where it is, but it should be in this range. The length of the variant, a complex variant type. If it's a complex variant, then you can combine different variants together. The chromosome copy number change type, some cytogenomic nomenclature for structural variants, and some RCGH ratio. Yeah, so a lot of components. Not every use case needs every component, but we defined all the components we could see which will be relevant or which were brought to us by actual use cases. So if you want to work with the IG, you take this variant and you constrain it to your needs. Maybe you need more components, maybe you just need 3 or 4 of them, then just constrain the rest to 0, 0 and you're good. Yeah, for the observation, the variants, we also have the implication of the variants. For these, we have a level of evidence. So what's the level of evidence of this implication? The related artifacts, which I already told you, points to the actual proof or the source, the paper, the journal article, the PubMed terms, whatever. Some secondary findings which are supporting this implication, and inherited pathogenicity for associated phenotypes and a mode of inheritance. Is it a dominant variant? Is it autosomal dominant? Stuff like that. Then we have the pharmacogenomics implications. There we have something like a mandatory medication which the patient should use based on some based on a physician in charge. Medication efficacy, medication metabolism, high-risk allele. High-risk allele would be an allele or a variation, which means that a patient has a high risk of a bad outcome if he takes this drug independently from metabolism or efficacy. Of course, medication usage. So for the recommended task, I can point to some medications and how they should be used in the recommended action. Yep. The somatic implications, this is the oncological field. I just told you they will be remodeled or further improved in the future, in the near future. I think for PGX it will be the next candidate to have another round to consolidate all the Steve profiles to make it easier for implementers. So we have the somatic diagnostic implication. How do the findings indicate the cancer? Then we have the prognostic implication. Do they imply positive or negative outcome for medication or therapy? So this is a purely patient-focused. So prognostic is really the prognosis for this patient. What are we expecting for this patient as an outcome? And then there's the predictive implication, which is more generic, which means if I have a subject, a patient with the following variants, and we are applying this medication to him, this should be responding to this treatment. We already had a look at this. Yeah, so this is for the more abstract UML overview of the IG. Next, I want to speak 1 or 2 sentences about our domain analysis model. Our DAM identified stakeholder use cases and workflows. Should have ongoing iterative feedback. We are currently focusing on the feedback of the profiles more than on the DAM. But yeah, the DAM is standard platform agnostic, human readable. It just captures what we are like to do independent of the technology we are doing it. It was the analysis of somatic acquired mutation, the cancer profiling goal. Determine individual mutation present, yes, no, which we are doing with the variant profile, tumor versus normal analysis. Yeah, so you can have a specimen which is the tumor or just blood from the patient if you're talking about rare diseases. It's not everything about oncology. I'm heavily biased, I'm sorry. genetic analysis for more accurate prognostics, and diagnose specific cancer subtypes and treatment options. Yeah, that's it for the DAM. I think I will move on with the actual examples so that we have enough time for some questions if there are some. Yeah, so this is actual FHIR code in XML. I'm sorry for the more modern people who are used to JSON. I'm still in XML. I can't help it. Example oncology report. We're starting with patient, practitioner, a specimen. This all is bundled together in a transaction bundle in our example, which helps you to put it onto your server. You just post your transaction bundle to your server, and you can have a whole example report with all the variants, the patient, the specimen, together on your server and can have a look. Yeah, so nothing fancy here, just a normal patient resource. There we have the practitioner and the specimen. Profile, but quite simple. So this is the first observation. It's a special observation. It's the tumor mutational burden. So this means how many somatic variants per megabase of sequenced DNA, because this has an impact on cancer treatment. If you have a lot of mutations inside of your tumor, you can benefit from that fact and treat the patient differently. So just a normal observation. We code everything in line. If we could do, this is a wrong code. I can see that. That's not a valid line code. But we have one for it. And here it was 25 somatic variants per megabase. By the way, this shouldn't be correct UCOM as well, I guess. I have to improve for the next slide. Then we have the oncology report. Inside this, in this report, we have the observations for the MSI here, the microsatellite instability. Yeah, line code here, wrong line code, I'm sorry. Value high. So still no variant. These are just more metadata about the patient variants. And now we are coming to the actual variants. Here we're having the, where are we starting? Yeah, it's observation. This is somatic predictive implication by computational analysis derived from some other resource. Level of evidence is here. This TBD, the TBD should be gone now. And it was based on the, let me have a look. No, that's the component, no, the level of evidence is expressed in the AMP guidelines. I'm sorry, this example doesn't make sense. There should be a value for the level of evidence here, not just AMP guidelines. We have the cancer type expressed here, and here's some medication code. Sorry for all the TBD. And here we have the actual overview of the diagnostic report, pointing to all the relevant resources, subject, performer, specimen, and then as results, tumor TMB, which we had a look at, MSI, SNV for JAK2 variant, KDR, RBB4. So these are the actual genetic variants pointed to in the observation. Then this is some therapy results down there. I guess it's some recommendations. And packed all together, this are some thousand lines of XML, which is a lot. It could be worse, but everything is structured, and it's a complex domain. It's not a problem of FHIR, I guess. It's a problem of cancer, or no, not of cancer, of genomics. That we have a lot of data to capture and to express. Yeah. Now for the last part, I want to show you some open source tools already adopting the IG. So the first one was the Smart Cancer Navigator done by the Boston Children's Hospital together with Harvard, I guess. Yeah, you can have a look at Smart Cancer Navigator GitHub.io. Unfortunately, it's not up to date. So we need an update on this tool. It's somewhere on my to-do list, but I don't know the time frame. to improve that. This was based on some STU2 version, unfortunately, so this has to be updated. There's the VCF2 FHIR project by OpenLMU. So this project takes a VCF file, because the question a lot of the time is, how do I get the FHIR report? Our labs, they don't speak FHIR, they speak PDF and fax, and maybe CSV. But all analysis pipelines are containing VCFs, of course. And if you have a VCF, the LMU built not a script, an application which takes the VCF file and transforms it into the IG diagnostic report, some observations, and there you could see G to A and A to G, G to A to G, heterozygous, the reference sequence, and the relation between the two variants is in cis. The gene ID is HNC 2621, which is CYP2C189, and the variant is present. So this is a handy tool if you are already having VCF files or access to VCF files. There you could start with variants. Of course, it doesn't capture everything because VCF doesn't capture everything, but at least the variants are good to go. Also a short slide about V2, which is developed by our Molit Institute. It's an open source virtual tumor board, progressive web app. We started with the focus on the web RTC video conferencing because our stakeholders were focused on web RTC video conferencing or on video conferencing in general. Then added some screen sharing, but we are now also displaying genomic reports. You saw that before in a smaller version. So these are FHIR variant observations read by our JavaScript-based PVA and displayed to the physician. It can also organize the conference and capture the recommendations which are the result of this tumor conference. Yeah, so this would be a screenshot how to capture the results and the recommendations, and there you have like, because of this variant, if you click here, you have a list where you could choose the relevant variant or variants, and these variants are leading us to this decision. Yeah, coming to the last slide. Review and close. Yeah, so I talked about FHIR Genomics Motivation significance. I showed you some tools, the DAM, our IG, which we developed. We had a look at the examples together, a brief look at tools, consuming our or adopting our IG. I welcome you to meet in code. I will be sitting around here all the dev days. If you have a question, always welcome. If you can't find me, ask questions on Sulip. Sulip has the benefit, I have to sleep. Sulip never sleeps, eats. Somebody is online all the time. This is the link to the Genomics stream. And join the web calls if you like to. So if you're involved in the genomic space, join our web calls, get yourself involved in Sulip. We're always loving feedback or new stakeholders presenting their use cases. So that's it for today from my side. Thank you so much. And if you got some questions, I think we have 2 minutes. No? We have no time for questions. Ask me during lunch. Thank you.